Our next presenter is Benjamin Chung. The topic of his presentation is SigB, a potential therapeutic target against the Staphylococcal small colony variants. PhD student from Science Department, Bachelor of Science Department. So Benjamin, anytime you're ready.
Okay, good morning everyone. Today I will be presenting on SigB, a potential therapeutic target against Staphylococcal small colony variants. So my agenda for today, I'll be talking about antibiotic and microbe stewardship, small colony variants, the SigB target, ongoing results, and concluding results. So antimicrobial misuse and overuse in humans, animals, and plants continues to be a global health crisis, leading to the rise of antimicrobial resistance. This is expected to cause as many as 10 million deaths annually by 2050 due to an increasingly inadequate, efficacious antimicrobials. And according to the World Health Organization, this is a global health crisis at all regions and income levels, and is expected to have an economic burden of $1 trillion by 2050. But this is not just a human health issue, but as one health and planetary health crisis as well, because we need to think about antibiotic resistance in animals as well as antimicrobial contamination in the environment. So the leading cause of antimicrobial resistance is antibiotics. So that is what I'll be focusing on today. And I also wanted to talk about one consequence of antibiotic overuse that is a bit less known to people than the usual antimicrobial resistance, and that is the formation of small colony variants. So what are small colony variants? Well, they are a subpopulation of wild-type bacteria that result in a phenotypic switch, resulting in slow growth and small colonies. This results in altered metabolic pathways and biochemical properties. And what's important is that there's a reduced expression of acute virulence factors. And what this means is that your body's immune system cannot respond to the bacteria because there's none of these acute virulence factors, and that's why SCVs are implicated in chronic persistent infections. So here's an example of Staphylococcus small colony variants. You can see from the circle, these are the small colony variants that are really tiny compared to the wild type colonies on the left, as well as altered biochemical properties resulting in a weaker gram staining of these small colony variants. There are three main virulence factors of small colony variants: significantly lowered antibiotic susceptibility, enhanced biofilm forming capacity, and enhanced intracellular host persistence. So all of these three factors contribute to why the infections caused by these SCVs are so persistent and long-lasting. They may even last for years. They are difficult to identify for diagnosis simply because they're so small, and no biochemical tests would just not detect them or result in inconclusive results. Yet, there is an expected 1 to 30% of these SCVs present in clinical samples. They are difficult to treat, and I'll explain it in the next slide. So if a patient comes in and presents with the wild-type infection symptoms, they're treated with antibiotics. These antibiotics can select for this SCV phenotype in vivo. So they will clear the symptoms, there's no more acute virulence factor formation, so they stop taking the antibiotics. And now these SCVs no longer are under the pressure of these antibiotics, will revert back to the wild type, and now you get back your whole vicious cycle of infection. There are veterinary implications of SCVs. They are implicated in chronic respiratory and cardiovascular diseases, and one of the main ones is chronic bovine mastitis. They are also implicated in clinical health situations such as foreign body-related infections, chronic bone infections, and about 29% are implicated in osteomyelitis infections, whereas 46% are implicated in cystic fibrosis-associated staphylococcal infections. So you can see in this image, this is a picture of a prosthetic knee replacement, and there was an abscess, and after isolating the abscess, they found that there were many small colony variants isolated. SCVs also have environmental implications. It is possible for there to be a proliferation of SCV reservoirs simply due to antibiotic overuse and environmental contamination. As such, therapeutic strategies for SCVs hold significance for planetary health across multisectoral settings. So how did I go about looking into how to target these SCVs? Well, first I looked into the formation of SCVs. There are many different stressors and pathways to the formation of SCVs, and one of them is intracellular induction without antibiotics, simply can just form within your body. The other is a variety of environmental stresses as listed here. On this picture here, this is an image of Staphylococcus that was subjected to cold stress for 3 months, resulting in the SCV phenotype after 3 months. So in this my research, I focus on antibiotic stress, which is a subset of the antimicrobials. This is a figure that I made that collated all the pathways and stressors that result in the formation of SCVs. So I'll be focusing on aminoglycosides of the antibiotics. You may be more familiar with the name gentamicin or other related antibiotics. And what research has shown is that induction by this specific pathway results in upregulation of SigB. So what is SigB? Well, it's an alternative sigma factor, and it's basically a global general stress response regulator. It has been linked to the induction of virulence of SCVs in ETC deficient types. What ETC means is electron transport chain. So these antibiotics select for mutations in the bacteria that disrupt this electron transport chain, which also may be why they exhibit different metabolic properties. So in this figure, you can see how the induction of staphylococcal SCVs result in the upregulation of SigB, and this is linked to increased biofilm formation, increased intracellular host persistence, and decreased antibiotic susceptibility. The three main virulence factors that I talked about in the first slide when I introduced SCV. So you can see how SigB is just globally linked to the whole virulence of SCVs. So my hypothesis is that targeting SigB will actually attenuate virulence in staphylococcal SCVs. So this is a general workflow of how my research is going to be, which I'll induce Staphylococcus to form SCVs by antibiotics. Then I'll target them with compounds targeting SigB, such as benzaldehyde derivatives, and I expect to see a decrease in biofilm forming capacity, increase in antibiotic susceptibility, as well as a lowered intracellular host persistence. And this will provide alternative strategies for antibiotic use and also help lower the cases of antimicrobial resistance. So my preliminary results, I have been working on a safranin biofilm assay currently, this allows me to take advantage of what's called a biofilm index, where I take the optical density of the biofilm biomass divided by the planktonic optical density, and this corrects for variations in growth time. And what this means is that I can compare wild type and SCVs despite them having completely different growth times. SCVs takes about 72 hours, a long period of time to grow, whereas wild type will grow within 24 hours. I'll also be looking at antibiotic susceptibility through microbial dilutions and disk diffusion assays. Another complementary study that we have been looking at is intracellular host persistence. This is where we take the bacteria and we internalize it with a specific cell line, let it grow for a designated period, then wash the extracellular bacteria off of an antibiotic wash. Then we can lyse the bacteria and plate to enumerate the colony-forming units. And by subjecting this to different conditions, we can see how good these compounds are at stopping the internalization of bacteria and persistence of bacteria within the host cells. Under this image, you can see just an immunocytochemistry staining of internalized SCVs in green. That's just a C spectrum. So antibiotic overuse and abuse continues to be a pervasive planetary health issue with the rise of antibiotic resistance, but another aspect of that is that it can also contribute to the formation of SCVs in veterinary, clinical, and environmental settings. In the One Health perspectives, we are all interconnected. All these three aspects of health are interconnected and will affect each other. So this challenge highlights the importance of resource stewardship of both antibiotics and microorganisms for the health of animals, humans, and plants. And I just want to conclude with a verse, Genesis 1:28. God blessed them and said to them, "Be fruitful and increase in number, fill the earth and subdue it, rule over the fish in the sea and the birds in the sky, and over every living creature that moves on the ground," to highlight resource stewardship. Thank you. Yes.
Okay.
So why did you choose, I guess, the SigB factor in particular? Because to my understanding, there's multiple genetic factors that are associated with SCV.
When I first was researching this project, I was actually just looking at wild-type biofilms in general, and SigB has just been implicated every single time in staphylococcal biofilm. So I was wondering, what's so special about this sigma factor? And the more I explored, the rabbit hole went deeper. It's linked to intracellular host persistence, it's linked to lower antibiotic susceptibility, and I realized, this is a really important target just in staphylococcus, especially with small grains. Yeah. Yes.
I just have I don't know what your plan is for knowledge translation activities, but I'm a nurse, so I don't practice anymore. I'm a climate and health educator, but from what your research will likely inform, are you planning on engaging with those who prescribe, because I think that if they were to have this information, that would be super useful for better health outcomes.
In fact, there are initiatives out there, I believe, that are trying to educate healthcare providers about these small colony variants simply because it's been missed so many times and just hard to diagnose there.
Yeah, thanks.
Sure.
Okay, great. Thank you very much.